Inside our body, we have many different types of ion channels. So, for instance, we have one type of ion channel we call the voltage-gated ion channel. And this is simply an ion channel that actually responds to changes in the voltage difference across the cell membrane. Now, many ion channels don't actually respond to changes in the voltage difference. Some ion channels, known as the ligand-gated ion channels, basically respond to the binding of a specific type of stimulating molecule we call a ligand. And when that ligand actually binds, it creates a conformational change that opens up that channel and allows the movement of the ions down their electrochemical gradient. Now, to demonstrate these ligand-gated channels and how they actually work, we're going to discuss one of the most well-studied ligand-gated ion channels we call acetylcholine receptors. And these are the receptors that are found on the membranes of postsynaptic cells found along neuron pathways. So, before we actually discuss the structure of this particular ligand-gated channel and how it actually works, let's remember a bit of biology about how action potentials actually move along neurons. So let's suppose we have a certain neuron and an action potential is generated on the axon hillock of that neuron. So as we generate it, that action potential begins to propagate along the membrane of the axon, and eventually it makes its way to the end of that axon to a location we call that axon terminal. So this is the axon terminal of this presynaptic nerve cell that generated that action potential. So, as the action potential makes its way to this axon terminal, within the cytoplasm portion of this axon terminal, we have hundreds of these vesicles, and each one of these vesicles carries many acetylcholine molecules. So, acetylcholine basically looks like this, and notice it contains a positive charge on this nitrogen, and what that means is it will be a polar molecule and it will be able to dissolve in the extracellular aqueous environment that exists between this axon terminal and the adjacent cell. So all of these vesicles are basically released via the process of exocytosis as a result of the stimulation of this action potential. And once we release these acetylcholine neurotransmitters, they basically diffuse across this area we call the synaptic cleft. And once the acetylcholine actually makes its way onto this side, it can now bind onto special sites, special regions, as we'll see in just a moment, on these acetylcholine receptors. So we have one here, one here, one here, and one here. So essentially, initially, before the binding process takes place, this acetylcholine receptor exists in its closed state. And in the closed state, these sodium ions and other ions, for example, potassium, cannot actually move across that cell membrane. But as soon as the binding takes place, that stimulates the opening of that channel, and these sodium ions can basically move into that cell, and that can depolarize the membrane and initiate an action potential on this adjacent cell. And so that's how action potentials move from one cell to an adjacent cell. So once again, acetylcholine receptors are ion channels that respond to the binding of special molecules we call neurotransmitters, and these are known as acetylcholine. They look like this. So when an action potential arrives at the terminal end of the presynaptic nerve cell, it stimulates the release of hundreds of these vesicles via the process of exocytosis, and each one of these vesicles carries many acetylcholine molecules. Now, the acetylcholine molecules then diffuse across this synaptic cleft and eventually make their way onto this cell membrane, and they bind onto these acetylcholine receptors. Now, the question is, how exactly does the binding process initiate the opening of that channel? So let's focus on the structure of acetylcholine. This is basically what it looks like. Acetylcholine is a pentamer, and what that means is it consists of 5 individual polypeptide chains. Now, 4 are actually different and 2 are identical. So we have 2 alpha polypeptide chains, 1 beta, 1 gamma, and 1 delta. So we have a delta shown in green. We have two of these alphas shown in blue. We have this light brown one shown by this. This is our gamma. And then this light purple one is our beta. And they basically form this pentamer structure. And it contains a hydrophobic region found within the membrane. It contains a region that protrudes out on the cytoplasm side. So this is the cytoplasm. And it contains a region that also extends out onto the extracellular side. So essentially this side is this side that points onto this extracellular environment, and this is the inside of this structure. Now, if we examine a top to bottom view of this structure, this is basically what we're going to see. And these are the two locations shown in red where acetylcholine molecules can actually bind to. So we have two different sites, and these sites exist on the boundaries between these two polypeptide chains and these two polypeptide chains. Now, notice that this is the channel that these ions can actually move through. This is that internal cavity. Now, before acetylcholine molecules can actually bind onto either one of these two regions, this cavity is essentially closed. So that is known as the closed state of acetylcholine receptors. So in the closed state, what happens is inside that cavity, we have these relatively large and hydrophobic amino acids. And these relatively large and hydrophobic amino acids basically restrict the size of the opening, and because they're hydrophobic, they prevent the movement of these polar positively charged ions, in this case, the sodium ion. So when acetylcholine is not bound to the channel exists in its closed state. In this state, the cavity of the channel contains large and hydrophobic amino acids, and these repel the polar ions and prevent them from actually passing across that cell membrane. So that's what we mean by the closed state of the ligand-gated ion channel. Now, what happens when acetylcholine actually binds to either one of these two boundaries, either the alpha-delta or the alpha-gamma boundary? Well, basically, once it binds, it creates a conformational change. And that conformational change involves the rotation of some of these membrane-spanning alpha helices that are found within that hydrophobic region. And when they rotate, when these alpha helices rotate, they essentially hide the large hydrophobic amino acids, and they expose small and polar amino acids. And that ultimately increases the size of that cavity and the size of the opening. And because we have the polar amino acids, now these polar ions, the sodium ions in this case, can easily move across and dissolve these cavity regions within the channels. So acetylcholine can bind at the alpha-delta or the alpha-gamma boundary regions shown here and here. Once it binds, it stimulates a conformational change that rotates the membrane-spanning alpha helices found within this hydrophobic core. This hides those large, bulky, and non-polar, so hydrophobic residues. At the same time, it exposes those small polar amino acids within that internal cavity. And this widens the entire cavity, and because now we have these polar amino acids, these ions can easily diffuse across that cavity of that channel, and this is known as the open state of the ligand-gated channel. So we conclude that unlike other types of channels, so the voltage-gated ion channels, which essentially open up as a result of a difference in voltage across that particular cell membrane, these don't actually respond to changes in voltage across that cell membrane. Instead, for these to actually open up and allow the movement of these ions down the electrochemical gradient, a specific type of molecule, in this case acetylcholine molecules, have to bind onto a special location and create that conformational change that opens up that cavity and allows the spontaneous movement of ions across that channel.